Hi, my name is Madison Audette, and today I'm going to talk about the enzyme maltase, and more specifically, a version of the enzyme maltase known as maltase glucoamylase. And the topic overview for today is we will first start by gaining a better understanding of the enzyme maltase. And to do so, we're going to understand what maltase is and its function. And then we'll understand where maltase is located and why it's important to us. And then we're going to take this a step even further, and we're going to look at a version of the maltase enzyme known as maltase glucoamylase. And in order to understand that enzyme, we will look at what maltase glucoamylase is and where it's found, what the structure and function of maltase glucoamylase is. Then we'll look at why it's important to us. And finally, we're going to understand some of the catalytic qualities of maltase glucoamylase. But first, we're going to look at what is maltase and why it's important to us and its function to us. Maltase is an enzyme that can be found in most organisms, including people, yeast, bacteria, and plants. The function of maltase is to catalyze the hydrolysis of the disaccharide maltose into molecules of alpha glucose, monosaccharides. In addition to this, maltase also plays a crucial function in our digestive systems. Its important role in digestion comes from when starch is assimilated in maltose via salivary or pancreatic enzymes. Once amylase is finished breaking down, the maltase enzymes transform the glucose. This is crucial as it's important in digestive function, as once glucose is present in the body, it can then be used for energy or stored in the liver as glycogen or animal starch. And below, we can kind of see a reaction scheme for the hydrolysis of the disaccharide into a monosaccharide. And now we're going to look at why this is important to us. Maltase, as mentioned in the previous slide, is crucial for the digestive systems of many organisms. Without maltase, many organisms' digestive systems would be impacted immensely. Maltase provides crucial assistance in the overall health of one's body and digestive system. Specifically, without maltase, we would be unable to break down maltose in order to produce glucose. And glucose, as previously mentioned, is essential as it is used for energy or storage in the liver as glycogen or animal starch. In addition to this, with the lack of maltase present or a deficiency in maltase, we would expect to see side effects such as stomach aches, indigestion, and acid reflux. This is because, again, the starches and sugars become more difficult for the body, and this in turn leads to other side effects and digestive issues. In turn, maltase should be thought of as how oil helps an engine run smoothly, as maltase helps the digestion of food. Now we'll look at the localization of maltase. As I've already mentioned in the previous slides, that maltase can be found in humans, plants, yeast, and bacteria. However, maltase is usually found within the brush border within the walls of the small intestine. Okay. Now we're going to look at the pathway of maltase, and we can kind of see over here in the diagram, we can see polysaccharide to disaccharide to monosaccharide. And so we start with starch and glycogen start in the mouth and the saliva. Amylase will break this down into the disaccharide known as maltose. Maltase is then found within the intestinal walls, and this will then break down maltose into glucose, which is a monosaccharide, via hydrolysis. And then we'll take the glucose right here, and it can be used in energy or stored in the liver as glycogen or animal starch. Okay, now that we've learned about maltase, we're going to take this even further, and we're going to look at maltase glucoamylase. What is maltase glucoamylase? Human maltase glucoamylase, MGM, is a small intestinal enzyme that works concurrently to hydrolyze the mixture of linear alpha-1,4 and branched alpha-1,6 oligosaccharide substrates that typically make up terminal starch digestion products. Where is it found? This is found in humans, and again, within the body, it is found within the brush border of the intestinal walls of humans. Why is this important to us? Okay, this is crucial to the hydrolysis of maltose into molecules of alpha glucose monosaccharides and the digestion of starches. And without this enzyme present, digestion would be altered and glucose production would be altered as well, which is crucial. As previously mentioned, it is used for energy or storage in the liver as glycogen or animal starch. Now that we know what it is, we're going to look even further at a structure. Using RCSB, I looked at a C-terminal subunit of human maltase glucoamylase, which the right you can see a blown up photo of that structure. And now we're going to take it further and look at the primary structure of this enzyme. So for the primary structure of a protein refers to amino acid sequence in a polypeptide. Using RCSB and the sample C-terminal subunit of human maltase glucoamylase structure, we can look at the primary structure, which can be seen in the image above to the left, that has a length of 1,857 amino acids. However, maltase has another molecule present, which is water, and the primary structure or amino acid sequence can also be seen above to the left. In addition to the primary structure sequence of maltase, above we can look at other properties of the primary structure, such as residues present and absent. The most important thing we can note here that is not present in this protein structure are non-standard residues. Non-standard residues refer to any residue that is not included in the list of standard residues. What we can note are residues that are present, which include aromatic rings and residues, buried residues, residues with clashes, accessible protein residues, and rings and residues. Now that we've looked at the primary structure, we'll look at the secondary structure. And the secondary structure becomes a little bit more complicated because we have turns and loops and alpha helices and beta helices. Specifically for MGAM, this primary structure defines this secondary structure, which has 114 beta strands, 48 helices, 23 turns, and anti-parallel beta sheets. Now that we've looked at the secondary structure, we will look at the tertiary structure. The tertiary structure of maltase determines the shape of its active site, which is complementary to a specific substrate. Some of the important qualities that we can note in this structure is that there's extra residues folded into an alpha helix and a loop in the active site. This forms positive 2 and positive 3 active subsites. We also can note the active site positions are at amino acid 529, 532, 1420, 1423, 1526. Another characteristic we can note about the tertiary structure is that it's globular and spherical in shape and it's water soluble. And the last thing is that there's nothing to note about the quaternary structure of MGM. Now we will look at the active site characteristics. Maltase glucoamylase has 2 active sites. The maltase site hydrolyzes terminal alpha-1,4 linked D-glucose residues from maltose or maltotriose, generating alpha-D-glucose. The glucoamylase site also has that alpha-1,4 hydrolase activity, but also hydrolyzes alpha-1,6 glycosidic linkages when they are adjacent to an alpha-1,4 linkage, liberating beta-D-glucose. Again, I also listed the active site positions. And then now we can note some of how this actually works. We can see the substrates will bind to the pocket via their non-reducing end, with a non-reducing sugar ring interacting with the buried -1 subsite and the reducing ring interacting with the surface exposed +1 subsite. Substrate cleavage occurs between -1 and +1 subsites and follows a catalytic mechanism, resulting in a net retention of configuration at the anoramic center. And to the left, we can kind of see a little diagram of how this all works with the active sites. Now we're going to look at an active site example with inhibition. So glucose amylase is an attractive target for inhibition by alpha-glucosidase inhibitors as a means of controlling blood glucose levels in individuals with type 2 diabetes. This is the most common inhibitor of MGM is arquebus, and it is the most widely used alpha-glucosidase inhibitor currently on the market and has been shown to be an efficient inhibitor of alpha amylase and of the C-terminal domains of MGAM, but a weaker inhibitor of the N-terminal domains of MGAM. And specifically, points in the structure, we can see that the residue ASP 1526 forms 1 hydrogen bond with atom N4B of arquebus, which is a candidate for an acid-base catalytic residue. And to the left, we can see chair conformation structures of arquebus and another inhibitor right below it and 2 other ones. Okay, and now we will look at some of the catalytic qualities. So the rates of maltase glucoamylase can be measured in several ways, but the most common way usually done is by running enzyme assays. An enzyme assay is quite simple. The steps can be seen below. In order to run an enzyme assay, you will mix enzyme and substrate. You will then record the rate of the substrate disappearance and or product formation as a function of time. You will then need to plot this in order to see the initial velocity for a substrate concentration. And to see how the rate changes, we will then change the substrate concentration and repeat. Usually alongside this, a glucose standard curve will be ran for each assay. And then the assays along with standard curves will then allow for Km, Vmax, and Kcat values to be calculated, which we can see in the next slide. So in this slide, to the left, we can see the Michaelis-Menten equation, and we can also see a graph of that. So using enzyme assays in the Michaelis-Menten equation, Km, Vmax, and Kcat values can be calculated. So specifically for MGAM, relative Km, Kcat, and Vmax values were as followed. 29.8 micromoles per minute, Km was 0.91 millimolar, and Kcat was 2.03 per minute. And these values will vary as they were found in a journal article, so it will vary depending on the experiment. Okay, and now we will look at what works and what doesn't work with MGM. Maltase glucoamylase works well with a mixture of linear alpha-1,4 and branched alpha-1,6 oligosaccharide substrates that typically make up terminal starch digestion products. The N-terminal catalytic domain of human MGAM has a preference for short linear alpha-1,4 oligosaccharides. So those two things are really important to note that really works with MGAM. But what doesn't work that we can know is pretty much anything but maltose. The specificity that maltase has for maltose is pretty high. So any substrate that pretty much is not maltose will not work with maltase. So some examples would be lactose or galactose. Lactose usually pairs with lactase. And now we can look at some of our concluding thoughts and key takeaways. So in conclusion, maltase glucoamylase is a complex enzyme that is crucial to body function, specifically our digestive system, and offers many structural and catalytic qualities that are crucial to many processes in the body. For the future, it would be important to further look at the absence of maltase glucoamylase and see how it impacts certain species, as we have a generalized idea of its impacts, but it would be beneficial to have supporting evidence. However, with the information we already have, now we can use this and apply it to other species, as all of the information found has mainly been from humans and mammals. We can also take relative Km and Kcat values and compare them to other literature, or even run our own experiment and compare our own values to relative values. And finally, here are my sources. Thank you for listening. Hope you have a good day.